Now let us discuss the various immunoglobulins. The immunoglobulins can be IgG, IgM, IgA, IgD and IgE. So the first immunoglobulin is IgG. So remember, the most abundant class of immunoglobulin, it's the IgG. It constitutes of 80% of total immunoglobulins in the blood. So it is the most abundant class in the serum and it is the major serum immunoglobulin. It is present in blood. It can be present in blood, plasma and tissue fluid. Remember, the immunoglobulin G, it contains fewer carbohydrates than the other immunoglobulins. The normal serum concentration is about 8 to 16 milligram per milliliter. The half-life, it is 23 days. So it has the longest of all. It is the longest of all the immunoglobulins. It has the half-life of around 23 days. IgG, it is the only immunoglobulin that can be transported through the placenta from the mother to the fetus. So that will provide the passive immunity to the newborn. So remember, the only immunoglobulin that is transported through placenta, it's the IgG. So it can activate the complement system through the classic pathway. So the IgG, it can activate the complement system through the classical pathway. So when the IgG antibodies, it binds to an antigen, they can trigger a cascade of complement proteins that leads to destruction of the pathogen. IgG, it can bind to the Fc receptors on the surface of phagocytic cells such as macrophages and neutrophils. So this process, it is known as opsonization. So it enhances the phagocytosis of the pathogen by these immune cells. So IgG, it has 4 subclasses, that is Ig1, Ig2, Ig3 and Ig4. So remember, Ig1, Ig3 and Ig4, they crosses the placenta and protects the fetus, whereas Ig3, it activates the complement. Remember, IgG, it is typically monomeric. That means it consists of single antibody unit, but occasionally it can exist in a polymerized form. So IgG, it is distributed between the intravascular component and the extravascular component. That means extravascular means inside the blood vessels and outside of the blood vessels of the body. So as the half-life of it's 23 days, that means it can remain in the bloodstream for an extended period of time. The breakdown and removal of IgG, it is unique that it varies with the serum concentration. So when the level of IgG, it's elevated in cases with the chronic malaria, Kala-Azar, the IgG synthesis against a particular antigen can be catabolized rapidly. So that this rapid catabolism that results in deficiency of specific antibody. In case of hypogammaglobulinemia, a condition that is caused by a low level of immunoglobulins, IgG provided for treatment will be catabolized more slowly. So that means it will remain in blood for extended period of time, so it remains in blood for the longer duration and hence it supports the immune system. The next immunoglobulin, it's the IgA. Remember, IgA, it's the second most serum immunoglobulin that constitute around 10 to 15% of the immunoglobulin. It has a short half-life of around 6 to 8 days. So IgA, it's mainly distributed in the secretions including the colostrum. That is the first milk that is produced by the mammary glands after the childbirth. It's seen in saliva, tears, sweat, and the wall of the intestine. So IgA, it occurs in two forms. The serum IgA and other is the secretory IgA. So IgA, it is found on the mucosal surface, and in secretion it exists as dimer that is formed by monomer. They are joined together at their carboxy terminal by a glycopeptide that is known as this J chain. So this dimeric form, it is known as the secretory component. So the secretory component, it contains a glycine rich polypeptide that is called secretory component or the secretory piece. So it is glycine rich polypeptide. So this component, it is produced by the mucosal or glandular epithelial cells that are involved in protecting IgA from denaturation by the bacterial proteases such as the intestinal mucosa. So secretory IgA, it is relative resistance to the digestive enzymes, and it is a reducing agent which make it effective in protecting the mucosal surface from the invading pathogens. The breast milk, which is rich in IgA, it will provide protection for the newborns during the first month of life. The transfer of maternal IgA antibodies through the breast milk helps to boost the infant's immunity. IgA, it does not fix the complement, but it can activate the alternate complement pathway. So there are two subclasses of IgA, that is IgA1 and IgA2. IgA2, it lacks the interchain disulfide bonds between the heavy and the light chain. The next immunoglobulin, it's the IgM. IgM, it is a pentameric consisting of 5 immunoglobulins. It consists of around 5 to 8% of the total serum immunoglobulins. So the molecular weight is around 9 lakh. So that is why it is known as millenary molecule. So which one is a millenary molecule? It's the IgM. So most of the IgM, it is found within the intravascular compartment. IgM, remember, it is considered as the oldest immunoglobulin class from the evolutionary perspective. It is the earliest synthesized immunoglobulin by the fetus in around 20 weeks of age. It cannot cross the placenta, and its presence in the fetus or newborn, it can indicate the intrauterine infections and making it a useful diagnostic in congenital infections like syphilis, rubella, and the HIV infections. So presence of IgM antibodies in serum of newborn would indicate congenital infection. IgM, they are relatively short-lived and disappear from the bloodstream earlier than the IgG. So presence of IgM in serum, it is indication of the recent infection. So which immunoglobulin that is produced in the primary response to antigen? It's the IgM. Which immunoglobulin is to be synthesized earliest by fetus? It's the IgM. Which immunoglobulin it's known as millenary molecule? It's the IgM. So IgM, it is significantly more effective than the IgG in the bacterial action, bacterial agglutination, and protection against the blood invasion by the microorganisms. So there are two subclasses of IgM, IgM1 and 2. The next is immunoglobulin D. Immunoglobulin D, it's present in the bloodstream at concentration about 3 mg per 100 ml. So half-life, it is around 3 days and it is mainly intravascular in distribution. So it is present on the surface of unstimulated beta B lymphocytes in the blood, and it acts as a recognition receptor for the antigen. So the binding of antigen to the cell membrane bound IgD, it activates the B cells, leading to its cloning and production of antibodies. So its main action is in the secondary response. So the main antibody in the secondary response is, remember, it's the IgD. It plays a role in opsonization the bacteria and making them easier to phagocytize. It crosses the placenta. It's of two types, IgD1 and IgD2. Immunoglobulin E, its half-life is 2 to 3. It is mainly produced in the lining of respiratory and intestinal tracts. Also known as reagent antibody. It mediates the type 1 hypersensitivity reaction that is responsible for asthma, hay fever, etc. So it cannot cross the placenta barrier. It is responsible for anaphylactic type of reaction. Also play important role in helminth parasites. So the IgG, remember, it protects the blood fluids. IgA protects the body surfaces. IgM protects the bloodstream. IgD mediates the reagent hypersensitivity. And IgE, its molecule on the surface of the B lymphocytes. Now, what are the abnormal immunoglobulins? The ones are the Bence-Jones proteins in the multiple myeloma. That you should know, Bence-Jones protein are seen in case with the multiple myeloma. Another important MCQ point of view. So it is typically found in the urine of patient with multiple myeloma, a type of blood cancer that is characterized by uncontrolled proliferation of the plasma cells. So the Bence-Jones proteins, they are uniform in nature, meaning they are composed of either kappa or lambda light chains, but not the both. So multiple myeloma can affect the plasma cells producing various types of immunoglobulins like IgG, IgA, IgD or IgE. So this condition, it is known as Waldenstrom macroglobulinemia, when it affects the IgM producing cells. So in Waldenstrom macroglobulinemia, excess production of myeloma proteins, that is the M protein, and they are associated with the light chains. The heavy chain disease, it is form of a paraproteinemia that is associated with lymphoid neoplasia, that is characteristic by characterized by overproduction of Fc, that is the fragment crystallizable part of the immunoglobulin. Other is the cryoglobulinemia. It is a condition in which the gel or precipitate forms in the blood serum when it is cooled but redissolves upon warming. So this condition can be seen in various diseases like myeloma, macroglobulinemia, and autoimmune conditions like SLE, etc.